In continuation to the previous lecture, today we will start finishing of enzymes. Enzymes are antigenic, and since problems occurred in the late 1960s when manufacturing workers exhibited severe allergic responses after breathing enzyme dust, procedures have now been implemented to reduce dust formation. These involve supplying enzymes as a liquid wherever possible or increasing the particle size of dry powders from 10 micrometer to 200 to 500 micrometer by either prelling. Prelling is a mixing the enzyme with polyethylene glycol and preparing a small sphere by atomization, or murmurizing. Murmurizing means mixing the enzyme with a binder and water, extruding long filaments, converting them into spheres in a murmurizer, drying them and covering them with a waxy coat. Industrial enzymology. Although many industrial processes such as cheese manufacturing have traditionally used impure enzyme sources, often from animals or plants, the development of much of modern industrial enzymology has gone hand in hand with the commercial exploitation of microbial enzymes. These were introduced to the West in around 1890s when the Japanese scientist Jokichi Takamine settled in the USA and set up an enzyme factory based on Japanese technology. The principal product was teca-diastase, a mixture of amylolytic and proteolytic enzymes prepared by cultivating the fungus Aspergillus oryzae on rice or wheat bran. Teca-diastase was marketed successfully in the USA as a digestive aid for the treatment of dyspepsia, which was then believed to result from the incomplete digestion of starch. Bacterial enzymes were developed in France by Auguste Biodin and Jean Efforant, who in 1930s found that Bacillus subtilis produced a heat-stable alpha amylase when grown in a liquid medium made by extraction of malt or grain. The enzyme was primarily used within the textile industry for the removal of the starch that protects the wrap in the manufacture of cotton. In around 1930s, it was found that fungal pectinases could be used in the preparation of fruit products. In subsequent years, several other hydrolases were developed and sold commercially. For example, cellulase lipase, but the technology was still fairly rudimentary. After World War II, the fermentation industry underwent rapid development as method for the production of antibiotics were developed. These methods were soon adopted for the production of enzymes. In the 1960s, glycoamylase was introduced as a means of hydrolyzing starch, replacing acid hydrolysis. Subsequently, in the 1960s and 1970s, proteases were incorporated into detergents, and then glucose isomerase was introduced to produce sweetening agents in the form of high fructose syrups. Since the 1990s, lipase have been incorporated into washing powders, and a variety of immobilized enzyme processes have been developed, many of which utilize intracellular enzymes. Currently, enzymes are used in 4 distinct fields of commerce and technology. First is industrial catalyst. Second is as a therapeutic agent. Third is as analytic reagents, and fourth is as manipulative tools, example in genetics. Of the thousands of different types of enzymes, about 95% are available from suppliers in the quantities ranging from microgram to kilogram, provided essentially for research purposes. Around 40 to 50 enzymes are produced on an industrial scale, that is ranging from multiple kilogram to tons per annum. The global enzyme market is currently dominated by the hydrolases, especially the proteases together with amylases, cellulases and lipases, supplied either as a liquid concentrates or as powders or granules that release the soluble enzyme on dissolution. Global production is dominated by two companies which between them supply more than two-thirds of the global enzyme market, namely the Danish company Novozymes with a market share of 47% and the US company DuPont which recently acquired Genencor with 21%.